Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on the biosynthesis of lipids, part 4. And this talk is an integral part of the paper Lipid Metabolism. The objective of the paper includes the understanding of the biosynthesis of isoprenoids and the significance or the implications of terpenoids biosynthesis. How do mevalonate and non-mevalonate pathway operates in a system? In this module of the biosynthesis of lipid, we study the synthesis of branch chain fatty acids. How these branch chain fatty acids are synthesized from the acetyl-CoA and how the branch chain alpha keto acid decarboxylase enzymes work to form the branch chain fatty acid. In addition to this, the fatty acid synthase system operation also be looked into. So continuing with the biosynthesis of lipid, part 2 explain these conundrums. To the aroma and the flavor of ginger, cloves and cinnamon, eucalyptus scent, red color of tomatoes, yellow color of sunflowers, they are of immense importance in folklore medicine. Camphor, citral, menthol are some of the well-known terpenoids bearing household importance and other useful uses. The other terpenoids includes curcuminoids present in turmeric and mustard seed, salvinorin A which is present in Salvia divinorum, cannabinoids present in cannabis species, bilobalide and gingolide present in Ginkgo biloba. In animals, the sterols and steroids are biologically created from the precursors of the terpenoids. Occasionally, terpenoids enhances the attachment of proteins to the cell membrane by a process called isoprenylation. The presence of terpenoids can be identified by performing Salkowski test. Structurally, terpenoids can be classified into different classes, namely hemiterpenoids, which has one isoprene unit and 5 number of carbon atoms. Monoterpenoids, having 2 isoprene unit, 10 number of carbon atoms. Sesquiterpenoids, bearing 3 isoprene unit, 15 number of carbon atoms. Diterpenoids with 4 isoprene units and 20 carbon atoms. Sesterterpenoids with 5 isoprene unit, 25 number of carbon atoms. Triterpenoids enclosing 6 isoprene unit with 30 carbon atoms, and tetraterpenoids with 8 isoprene units having 40 carbon moieties. In addition to these classes, polyterpenoids and meroterpenoids are also present. The polyterpenoids possess a greater number of isoprene units, whereas meroterpenes are the compounds with a partial terpenoid structure. The isoprenoids, which are often called as terpenoids, are the organic chemicals, especially the hydrocarbons. The chemicals which are composed of hydrogen as well as carbon atoms. As name indicates, the isoprenoids or terpenoids especially evolved from 5 carbon isoprene units. CH2C, CH3, CH double bond CH2. These isoprenoids differ in their basic carbon skeletons as well as in functional groups. These lipids type, the isoprenoids or terpenoids, contributes the biggest group of natural products. Close to the 60% of the natural products belongs to this category, and they are present in each and every class of living thing. The terpenoids isolated from the plants are being used comprehensively for their aromatic traits. They provide aroma, the fragrance, and they play a vital role in the traditional herbal remedies. Besides possessing the antimicrobial, antineoplastic, and other additional pharmaceutical utility. The mevalonate pathway feedback and regulation. A number of key enzymes can be activated in the course of DNA transcriptional regulation. On the commencement of SREP-P1 and SREP-P2, which are sterol regulatory element binding protein, both the isoforms SREP-P1 and SREP-P2 acting as an intracellular signals perceive low cholesterol levels and kindle endogenous production by the HMG-CoA reductase pathway through lipoprotein uptake increase and LDL receptor upregulation. The regulation of mevalonate pathway is also accomplished by scheming the translational rate of messenger RNA, reductase degradation, and phosphorylation. A number of drugs, including statins. Statins are the class of cholesterol-lowering drugs that targets the mevalonate pathway. And atorvastatin, which is also known as Lipitor, inhibits the HMG-CoA reductase step. Bisphosphonates, another class of drugs targeting mevalonate pathway, is used to take care of diverse bone degenerative disease. The mevalonate kinase deficiency results in hyperimmunoglobulinemia D syndrome, abbreviately known as HIDS, and mevalonic aciduria that affects the mevalonate pathway. The non-mevalonate or the alternative pathway. In most of the bacteria and some protozoans, including the malarial parasites and plants, isoprenoids are produced by using a non-mevalonate or alternative pathway called as the methyl erythritol phosphate pathway or the MEP pathway. The production of both mevalonate and non-mevalonate pathway are isopentenyl pyrophosphate and dimethylallyl pyrophosphate. But the enzymatic reactions involved in both mevalonate and non-mevalonate pathway for the conversion of acetyl-CoA to isopentenyl pyrophosphate are utterly different. In higher plants, the mevalonate pathway maneuvers in the cytosol, while the non-mevalonate or MEP pathway drives in the plastids. Among the bacteria, the MEP pathway operates in E. coli and Mycobacterium tuberculosis. The non-mevalonate pathway or the MEP pathway, also known as 2-carbon methyl d-erythritol 4-phosphate or 1-deoxy-D-xylulose 5-phosphate pathway or mevalonic acid independent pathway, occurs in many bacteria, apicocomplexan of protozoans, as well as in the plastids of plants. In 1980s, the pathway was discovered whose enzymatic steps are as follows. The mevalonate pathway. In archaebacteria as well as in eukaryotes, the mevalonate pathway begins in the similar way as the acetyl-CoA is the feedstock of the pathway. There are two parts of the mevalonate pathway, the upper part of the mevalonate pathway as well as the lower part of the mevalonate pathway. The steps of the upper mevalonate pathway includes 3 reactions. The 2 molecules of acetyl-CoA yield an acetoacetyl-CoA like in cholesterol biosynthesis or steroid biosynthesis. Then further in the second step of the reaction, the condensation of acetoacetyl-CoA to form 3-hydroxy 3-methyl-glutaryl-CoA or the HMG-CoA. The third step of the reaction includes the reduction process where this HMG-CoA gets reduced to form mevalonate. all these three steps are similar to that of the steroid metabolism as well as the cholesterol metabolism. So these three steps constitute the upper mevalonate pathway, which resembles the steroidal as well as cholesterol biosynthetic pathway, whereas in the lower mevalonate pathway, conversion of mevalonate to IPP and DMAP has three reported variants. The mevalonate of eukaryotes is phosphorylated at 5-hydroxyl position twice, followed by decarboxylation to yield isopentenyl pyrophosphate, which is the first variant. The second variant is the mevalonate of archaea, for example, Halopharax vulcani. It is phosphorylated 5-hydroxyl position followed by the decarboxylation to yield isopentenyl, and this again phosphorylated to get isopentenyl pyrophosphate. So there is an addition of one step in between in the second variant, which is an archaeal mevalonate pathway 1. The third variant has been observed in Thermoplasma acidophyllum. The mevalonate is phosphorylated where the 3-hydroxyl group is phosphorylated followed by 5-hydroxyl position to produce mevalonate 3,5-bisphosphate. So a new variant is formed, and this mevalonate 3,5-bisphosphate is then decarboxylated to isopentenyl and further followed by the formation of IPP or the isopentenyl pyrophosphate. This results in an archaeal mevalonate pathway 2. The isopentenyl pyrophosphate and dimethylallyl pyrophosphate are not only the end products of mevalonate and non-mevalonate pathways, but also they serve as the precursors of isoprene units, 5-carbon or monoterpenoids having 10-carbon, diterpenoids 20-carbon, carotenoids 40-carbon, plastoquinone and chlorophylls having 45-carbon. The synthesis of the entire higher terpenoids ensued by the configuration of geranyl pyrophosphate, farnesyl pyrophosphate and geranyl pyrophosphate. Even though both the mevalonate and the non-mevalonate or the MEP pathways are reciprocally elite in the majority of the organism, relations amid them have been testimonied in few bacterial species and plant species as well. The mevalonate or HMG-CoA reductase pathway is a classical and an imperative metabolic pathway present in many bacteria and higher eukaryotes. It is vital for isopentenyl pyrophosphate and dimethylallyl pyrophosphate production, and dole out as the source for the biosynthesis of molecules employed in N-glycosylation and protein prenylation, maintenance of cell membrane, as well as in protein anchoring. In contrast to the classical metabolinate pathway, most of the bacteria, some of the protozoans, and plants fabricate their isoprenoids using the MEP or the non-mevalonate pathway. As we can see in the following table, the pathway operated both the mevalonate as well as non-mevalonate, which is MEP pathway, and the organism in which these pathways were carried out can be well studied and understood. Like mevalonate pathway is present in archaea, fungi, as well as in animals. The non-mevalonate pathway or the MEP pathway is present in green algae and protozoans. However, both mevalonate and non-mevalonate pathway has been seen to be operated in bacteria as well as in plants. Among the non-mevalonate pathway or deoxyxylulose pyrophosphates pathway, the DOXP synthase enzyme converts pyruvate and glyceraldehyde 3-phosphate to 1-deoxy-D-xylulose 5-phosphate. The pyruvate formation is results from the glycolysis, as well as glyceraldehyde 3-phosphate also comes through glycolytic reactions, which is utilized by DOXP synthase to produce 1-deoxy-D-xylulose 5-phosphate. In the second step of the reaction of non-mevalonate pathway, reduction process takes place in presence of enzyme DOXP reductase that forms 2-carbon methyl D-erythritol 4-phosphate, or also called as MEP. The 2-carbon methyl D-erythritol 2,4-cyclopyrophosphate or MECPP is produced due to the succeeding 3 reaction step, which is catalyzed by 4-diphosphocytidyl 2-carbon methyl D-erythritol synthase. This 4-diphosphocytidyl 2-carbon methyl D-erythritol kinase and 2-carbon methyl D-erythritol 2,4-cyclodiphosphate synthase, all these 3 enzymes catalyze the succeeding reaction of MECPP. In the final steps, this HMBB PP synthase converts MECPP to 4-hydroxy 3-methyl but 2-vinyl pyrophosphate, and this further reduced to isopentenyl pyrophosphate and DMAPP. This DOXP reductoisomerase enzyme, which is a key enzyme of non-mevalonate pathway, is specifically inhibited by phosmidomycin, an important antimicrobial compound. And this inhibition of this enzyme consequently signifies that this is an attractive target for antibiotics as well as some of the antimalarial drugs. The HMBPP, which is an intermediary of the MEP or DOXP pathway or the non-mevalonate pathway, activates some of the human cells, T cells, which are major population of the peripheral blood of humans. So students, today we have learned about the biosynthesis of isoprenoids or the terpenoids, which have been evolved from the 5-carbon isoprene units. Also, there are two pathway operates, the mevalonate and the non-mevalonate pathway. We have discussed about the MEP and DOXP pathway and its significance. In the coming lectures, we will continue with the biosynthesis part of lipids. Thank you.